Hello everyone, welcome to Master Microbiology. In this video today, I'm sharing with you common terms in gram-positive and gram-negative bacteria. The common terminologies that are used in the gram-negative and in the gram-positive bacteria. Now, I will start with the general terms, and that is what we mean, that is the gram stain. The gram stain is a differential staining method or procedure or technique used to classify bacteria into gram-positive, that is purple color at the end, and gram-negative, that is pink to red color at the end based on their cell composition. The next thing I want to talk about is peptidoglycan, also referred to as murine. This is a polymer that forms a rigid structure in bacterial cell walls. Gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thinner layer. Number three is cell envelope. The cell envelope is the structure surrounding the bacterial cytoplasm, including the cell membrane and cell wall. In gram-negative bacteria, it also includes an outer membrane. Now, let's talk about teichoic acids. These are polymers that are present in the cell wall of gram-positive bacteria. They play a role in maintaining cell shape, regulating ion exchange, and promoting adhesion. Now, let's talk about lipoteichoic acids, that is LTA, lipoteichoic acid. The teichoic acids anchored to the lipid membrane in gram-positive bacteria. So lipo has to do with lipid. So you can use that to differentiate it. So these are teichoic acids that are anchored to the lipid membrane in gram-positive bacteria. They are involved in host immune interaction. What then do we mean by outer membrane? Outer membranes are found in gram-negative bacteria. This lipid bilayer contains lipopolysaccharides, LPS, which provide structural integrity and protection against antibiotics. Now, lipopolysaccharides is the next term we want to define. Lipopolysaccharides are also known as endotoxins. Lipopolysaccharides are found in outer membrane of gram-negative bacteria and can trigger strong immune responses in hosts. Number 8 is periplasmic space. This is the space between the inner, that is the cytoplasmic and outer membranes in gram-negative bacteria. It contains enzymes and transport proteins. Now let's talk about porins. Porins, P-O-R-I-N-S, porins. These are protein channels in the outer membrane of gram-negative bacteria that allow the passive diffusion of small molecules. Endotoxin. Endotoxin is a toxic component of the lipopolysaccharides in gram-negative bacteria that can cause severe inflammatory responses in the host. Exotoxin. These are toxins secreted by both gram-positive and gram-negative bacteria, often causing specific tissue damage. Now, the following terms I want to talk about is strictly related to gram-positive bacteria alone, and that is thick peptidoglycan layer. So gram-positive bacteria have a significantly thicker peptidoglycan layer, 20 to 80 nanomicron, compared to gram-negative bacteria. Now, let's talk about crystal violet retention. Gram-positive bacteria retain the crystal violet iodine complex during gram staining due to their thick peptidoglycan layer. Mycolic acids, MyColic. Long fatty acids found in the cell walls of certain gram-positive bacteria, such as Mycobacterium species. S layer, also referred to as surface layer. This is a protective layer of proteins or glycoproteins found outside the peptidoglycan in some gram-positive bacteria. Spores. These are dormant, highly resistant structures formed by some gram-positive bacteria, e.g. Bacillus and Clostridium, under adverse conditions. Now, the following terms I am going to mention now are related to gram-negative bacteria strictly. Number one is thin peptidoglycan layer. In the case of gram-positive bacteria, they have thick peptidoglycan layer, but gram-negative bacteria have thin peptidoglycan layer. So the peptidoglycan layer in gram-negative bacteria is much thinner, and the measure is 2 to 7 nanomicron, and they are thinner than in gram-positive bacteria. The next thing we want to talk about is safranin retention. Gram-negative bacteria appear pink after gram staining because they do not retain the crystal violet stain and are counterstained by safranin. Brown's lipoprotein. Brown's lipoprotein is a protein that links the outer membrane to the peptidoglycan layer in gram-negative bacteria. Now, let's talk about efflux pumps. Efflux pumps are protein transport systems in gram-negative bacteria that expel toxic substances, including antibiotics, contributing to resistance. O antigen. O antigen is a component of the lipopolysaccharides in gram-negative bacteria involved in immune evasion and serotyping. Secretion systems. Secretion systems are complex protein transport systems, e.g. type 1, type 4, type 1 to type 6 secretion systems used by gram-negative bacteria to deliver effector proteins into host cells. Now, the following are comparative terms that are linked to both gram-positive and gram-negative bacteria. And the first comparative term is antibiotic sensitivity. Gram-positive bacteria are generally more sensitive to antibiotics than that target peptidoglycan synthesis, e.g. penicillin, while gram-negative bacteria are often resistant due to their outer membrane. Gram reaction. The outcome of the gram stain procedure distinguishing bacteria based on cell wall structure. Pathogenicity. Gram-negative bacteria are often more resistant to host defenses due to their outer membrane and lipopolysaccharides, while gram-positive bacteria can produce more potent exotoxins. Capsule. Capsule is a polysaccharide layer surrounding some gram-positive and gram-negative bacteria, aiding in immune evasion and adhesion. Number 5 in this segment that we are comparatively looking at is flagellar arrangement. Both gram-positive and gram-negative bacteria can have flagella, but the basal body structure differs due to variations in the cell envelope. These terms are foundational for understanding the structural and functional differences between gram-positive and gram-negative bacteria, as well as their roles in pathogenesis, ecology, and response to treatment. So this is where I'm going to stop on these common terms related to gram-positive and gram-negative bacteria. This is Master Microbiology YouTube channel. I want to subscribe to this YouTube channel, share the link of this video, like this video, and if you have comment and question, go ahead in the comment section and drop your comment and question there. Now, before I go, let me ask you this question. Do you have any special question you want to ask me and you just want it one-on-one? You don't want to ask it on this group platform in the comment section? Then I'm available to take your question on a personal level and to assist you. Now, what you need to do is dial the number +234 80 36. I take that again. +234 80 369 10988. Dr. Olubenga Adetitun is my name, and on this channel I bring you information together with others on microbiology issues. Why? So that you can master microbiology. This video that's showing up from this Master Microbiology YouTube channel is the next you need to watch now. Go ahead, click it, and watch it. I will see you in the next one. Until I come your way again, bye for now.